Hi, I'm going to be recording chapter 4, and I will keep it as brief as possible. Like I said, there are some few things I will mention, and there are some things I will not mention. I will not have to teach. Now, the things I'm not teaching, it doesn't mean they are not relevant, it doesn't mean they are not going to be important, and it doesn't mean it will not be on the test. It only means that they are easy to read. All right. Now, this is cell structure. So principally, we'll be looking at cells. We'll be looking at the types of cell. We'll be looking at different organelles in the cell. And so it's pretty straight and it's simple to study. All right. So all cells are composed of all living organisms are composed of cell. And like mentioned yesterday while I was recording chapter 3, that the cell is a structural and functional unit of life. What that means is this: there is nothing smaller than a cell will still be functional as a living thing. And structurally too, nothing smaller than a cell will be structurally classified as a living thing. So the smallest thing that can be classified as a living thing is a cell. The basic unit of life, all organisms, however big or however small, called organisms are made up of cells. So if anything is called organism, it is made up of cell. Your pets are made up of cell. You are made up of cell. Bunnies are made up of cells. Everything that is living have cells. Now, new cells arise from pre-existing cells. There is no cell that exists by itself. So I can't be talking more about that, but the principle of cell is that cells are able to reproduce. They're able to regenerate. Now, we have cells that can differentiate into different type of cells. So when the cell arises, it's from pre-existing cells. All right. And the general concept of that is that you have cells that are called germ cells in the body. They are cells that are like raw material cells. They are used to make other cells, right? You know, in your cell body, you have somatic cells, you have reproductive cells. All right. Now, prokaryotic cells evolved first, and I remember I mentioned this in one of the videos. Prokaryotic cell evolved 1 billion years before eukaryotic cell ever existed, because the environment was not conducive for eukaryotic cell to survive. So prokaryotic cell they are resilient. They are more resilient. Like, there was no oxygen at the start of the world, so there was no eukaryotic cell they can't survive in such environment. So prokaryotic cells are able to survive. So they were in existence about 1 billion years before before eukaryotic cells came into existence. So two fundamentally different cells that we have the eukaryotic cells and we have the prokaryotic cells. And if you remember, when I told you about domain is BAE, B-A-E, I told you that we have 3 domains of classification. We have the bacteria, the archaea, then we have the eukarya. And so this class belongs to the eukarya, the class. So you have the eukarya here. Sorry, this is not going to be good. Okay, so we have this class. Bacteria and archaea belongs to the prokaryotic, and the eukaryote belongs to the eukaryotic cell. So eukaryote belongs to the eukaryotic cells. Now, the major difference between eukaryotic cells and prokaryotic cells is this. Now, I know you will have heard over time that eukaryotic cells have nucleus and prokaryotic cell does not have nucleus. Then you go ahead and they also told you that prokaryotic cells have DNA, which is also present in DNA in eukaryotic cell. And sometimes you may get confused, like, how do they have DNA and they don't have a nucleus? Now, this is my definition of it. You can have this bottle and not have water, right? So, but the other way I will explain it that makes it easier for me or for my students is this. This is a wallet. And if I ask you, do you have a wallet? It's not everybody that have a wallet. But what do we put in a wallet? We put our driver license. We put our cards in the wallet. Then sometimes you will want to put some cash in the wallet as well. Now ask me, do you have a card with you there? Yes, as I'm talking to you now, most likely you're going to have a card with you. Now that tells me that even though you don't have the wallet, you have the content of the wallet. It's the same thing with eukaryotic cells and prokaryotic cells. Prokaryotic cells does not have the wallet, but they have the content of the wallet. So this is it. Prokaryotic cell does not have the nucleus, which is the nuclear membrane, the covering, the membrane that tells that the nucleus is present. The nucleus have a double membrane just like the plasma membrane. Now, eukaryotic cells have the plasma membrane, the nuclear membrane. They have what we call an enclosed nucleus. But prokaryotic cell does not have the membrane, they don't have the enclosed membrane nucleus, but they have the DNA. Now, there is no cell that can exist without DNA. So, prokaryotic cells have the DNA, like you can see here. You will notice that, I'm getting my, all right, good. You will notice here you can see all these strands. That is the DNA. The DNA is present in the prokaryotic cell. And here you see the nucleus. And you see the DNA inside the nucleus. So they both have the DNA. Don't confuse it. DNA and nucleus is not the same. The DNA is resident inside the nucleus. There are other things there. But the DNA is resident inside the nucleus, which is the most important thing in eukaryotic cell. But prokaryotic cell doesn't have the DNA, so, but they still have, doesn't have the nucleus, but they still have the DNA. Good. All right, so that's why we say here that they lack a membrane bound nucleus, because they actually have the DNA. They have the DNA, but they don't have a wallet, but they have the content of the wallet. So think about it. Both of them have the content of the wallet, but one of them have the wallet, the other doesn't have it. So eukaryotic cell have nucleus, prokaryotic cell doesn't have a nucleus. It doesn't have a membrane bound nucleus, but they both have DNA. Again, there's no cell that can exist without a DNA. Now, prokaryotic cells are smaller than eukaryotic cells. That is basic. Eukaryotic cells are larger. Looking at this, you can tell they are larger than prokaryotic cells. Then in eukaryotic cell, you have one domain, which is the eukarya, and prokaryotic cell, you have the two domains, which is bacteria and archaea. Another thing you may want to look at about the prokaryotic cells is that, like I said, eukaryotic cells have membrane-bound nucleus. That tells you that they have other membrane-bound organelles. Look at it. You can see other membrane-bound organelles right here. You can see other membrane-bound organelles. So you can see the rough endoplasmic reticulum. You can see the smooth endoplasmic reticulum. You can see the Golgi apparatus. You can see the mitochondria. All of them, they have membrane-bound organelles. But you won't see that in prokaryotic cells. So eukaryotic cell is more organized, is larger, is more complex, whereas prokaryotic cells are simpler and less complex. All right, so component of prokaryotic cells. What are the things you see in prokaryotic cells? Now, prokaryotic cell, like I said, all cells have DNA. Now, but they don't have a nucleus. They don't have a nuclear membrane, but they have a location. Now, when you come to class, while we're still on campus, you don't have a room to yourself in the class, but you have a region where you sit, your location where you sit, and that is your location at that time, is your region, but it's not a room like you have at home. So, prokaryotic cell, even though they don't have a nuclear membrane that surround their nucleus, there's a location where that surround their DNA, there's a location where the DNA is resident. The location where the DNA is resident is called the nucleoid region. So sometimes we just put region. So it's called the nucleoid region. So you see it as nucleoid region. Sometimes you see it as nucleoid. It's still the same. It's just saying that this is the nucleoid region. The location of the DNA is what we call the nucleoid region. It's usually at the center, centralized like that. All right. Then it has a ribosome. Now, there's no cell that can also exist without ribosome. All cells need protein to function. I told you yesterday that the machinery that does every work in the body is not the carbohydrate, it's the proteins. Protein does the work, carbohydrate provide energy to get the work done. No protein, nothing does the work. Energy does not do the work. There is something that is powered. If you have a car, if you have gas and you don't have a car, you can't move to another location with gas alone. That is the way carbohydrate is. Carbohydrate is gas. Protein is a car, is the engine, is a car that moves you around. You just need carbohydrate to perform, for protein to perform its function. So every cell, ribosome is what makes the protein, and so every cell needs ribosome. Every cell also needs cell membrane. There's this current study that have shown that a cell can still exist without the nucleus for some moment. But the moment the cell membrane is cracked, the cell dies. So that means the cell membrane is actually the protective, is what sustains the cell, not the nucleus as it was earlier thought. The heart of the cell seems now to be the cell membrane that protects the cell and not the nucleus. So cell membrane is very important. It determines what goes in and out of the cell. It protects the cell. It helps with a lot of so it must be present in every prokaryotic cell. Even though they are smaller, they must have that. Now, prokaryotic cell also have the cell wall as well. You can see that there. Now, there are other structures there, but those are the major that must be in all prokaryotic cells. Now, eukaryotic cells. Eukaryotic cells contain specialized organelles, and that's the key thing about them. You have many organelles in eukaryotic cells that are membrane bound. So we say that they are compartmentalized into organelles, and these organelles perform specific functions, and they are usually membrane bound organelles. This domain is the eukarya, and it consists of protists, plants, fungi, and animals. So you can think of it as PPFA, or you can think about it as FAP, which is FAPP, or you can think about it as PAF, PPAF. Either way you want to remember it, that'll be fine, but you just need to know those basic things. I tell my students sometimes that there are some basic things you should know when you finish a course. It's not because you made an A or a B in the course, but by knowing that you have gone through that course, there are some things you should know. And these are basic things you should never forget since you have taken this course. You should know the domain is BAE, B-A-E, bacteria, archaea, and eukarya. You should know that archaea and bacteria and archaea are the prokaryotics. You should know that eukarya are the eukaryotics. All right. Now, this is the nucleus here All right, so we have a lot of membrane-bound organelles. You can see the nucleus here. It has membrane. So membrane-bound organelles are organelles that have membrane, even though they are inside the cell. They still have their own membrane. All right, so you can see perizysosome, you can see endoplasmic reticulum, you can see the mitochondria, you can see the nucleus. All of them have membranes, and they have features. Now, there's something we call the endomembrane system. You may just want to know that the endomembrane system, when the nucleus, there are some ribosomes that are stored on the nuclear membrane. So those ribosomes, they also make proteins. Now, once they make their protein, you know, the rough endoplasmic reticulum is a continuation of the cell membrane, of the nuclear membrane. So then you have the smooth endoplasmic reticulum as well, right? So there are some, if you look at that cycle, you have the nuclear membrane continues with the endoplasmic, with the rough endoplasmic reticulum. Then you have the Golgi apparatus that also help to process proteins. Then you have vesicles. Then you have the cell membrane that some proteins can be packaged and go out of the cell through the cell membrane. So when you consider that transportation mechanism, you think about the nuclear membrane, rough endoplasmic reticulum, Golgi apparatus, some vesicles, then the cell membrane as endomembrane system. Now, principally, just to note here, there are some proteins that are made by some ribosome and they will be used by the cell. And there are some proteins that are made by some ribosome and will need to be transported to other cells. Now, the endomembrane system take care of such. The proteins that are the ribosomes that are on the on the nuclear membrane or on the rough endoplasmic reticulum, the proteins they make goes through that transport system and they may be sent out of the cell. But there are some other ribosomes that are just in the cytoplasm. We call that the free ribosome. Those ribosomes make proteins and the proteins will not be transported out of the cell. So those proteins will be for the cell purpose. It will be used for that cell activity. So we have two types of ribosome. Now, two types based on their location. Some are just in the cytoplasm. They are free ribosome. They are free to move in the cytoplasm. We call them free ribosome or unbound ribosome. Then the ones that are stored like pimple on the plasma membrane, on the nuclear membrane or on the endoplasmic reticulum, the rough endoplasmic reticulum, we call those ones bound ribosome. Bound in that B-O-U-N-D, bound in that they are not free. They are stored or stocked on the membrane. All right, so the functions of all of these organelles are here. Please look them up and principally anywhere you see ribosome, it makes protein. Just make it a structure. Anywhere you see ribosome, it makes protein. So if ribosome is on the rough endoplasmic reticulum, which is why it is rough, that means the rough endoplasmic reticulum makes protein. The smooth endoplasmic reticulum does not have ribosome. That means it doesn't make protein. It makes lipid. All right, this is animal cell and this is plant cell. You should be able to identify them, look at their shape, their different shapes. Then majorly, something is unique. Animal cell, plant cell have central vacuole, which is what maintains help them to maintain pressure against the cell wall, which I will be talking about hopefully when I'm talking about osmosis. I will discuss that clearly, but this is what this is a major difference between the plant cell and animal cell. Plant cell have central vacuole, so with that they are able to store a lot of water. Animal cell does not store water as such. They have tiny vacuoles. Now, so if you put animal cell in water, the animal cell will take in a lot of water, and there's no central vacuole to accommodate this pressure. But if you put plant cell in water, plant cell takes in water, but it's fine because it has a central vacuole. I'll be talking more about that. Talking about hypotonic, hypertonic, plasmolysis, crenation. We'll be talking about that later, but you don't need it in this chapter. All right, so this is the plasma membrane. I've talked about the plasma membrane in the previous chapter, so I won't be focusing on that again. But you should know that you have the plasma membrane. It controls the passage of organic molecules, ions, water, oxygen that comes in and out of the cell. And I've told you in the previous chapter that there are two layers, right? It has two layers. It has this first layer and the second layer that are impacted to adapt to their environment. And you have glycoproteins there. That is protein that are associated with carbohydrate. You have glycolipid, protein that are lipids that are also mixed with carbohydrate. You have some proteins that are on the peripheral. Now, peripheral protein means that they are on the surface. And we also have some protein that are integral. It means they are integrated. And so you can see this protein is integrated. It passed through the whole length of the cell membrane. That means it is integrated. This is also going to be an integrated protein, even though it is a channel, which we'll be talking about when we are talking about transport system. So we have integrated protein, we have peripheral protein, we have the filament, cholesterol, which is what gives the cell membrane its shape. All right, so let's go. Now, there is microvilli on the plasma membrane, and the purpose of the microvilli is to increase the absorbing surface, right? Is to increase the absorbing surface. That's majorly what it does. I'll just leave that. Now, cytoplasm. Now, cytoplasm is key. Cytoplasm is key. Now, there is the cell membrane. Inside the cell membrane, at the center of the cell, you have the nucleus. Everything after the nucleus and the cell membrane is what we call cytoplasm. Now, there's a jelly-like structure, liquid inside the cytoplasm. That is what we call the cytosol. Now, the cytosol is what gives a little structure to the cell membrane or organization. Now, organelles are all in cell membrane: ribosome, mitochondria, Golgi apparatus, vacuoles. They are all in the cell membrane. Now, the reason they have a little measure of organization is because of the cytosol that positions them in the likely position that they are supposed to be. So, this is cytosol. It is a liquid, a jelly-like liquid inside the cytoplasm. All right. Then the cytoplasm is the space in between the nucleus and the cell membrane. Now, this consists about 70% of water, but it has a semi-solid consistency. All right, let's go. There's something I didn't mention about plant cell and animal cell the other time. Key thing about plant cell and animal cell, the difference is that I've talked about central vacuole, yes. You also remember that plant cell have chloroplast, and chloroplast is a key difference, is a very key difference. That's why they have their color. That's why they undergo photosynthesis. Plants, human beings, animals don't undergo photosynthesis because chloroplast is absent, right? Now, Okay, so I think that's what so let's go to the nucleus. Now, I've talked a little about the nucleus, but I will just go through a few things. Now, inside the nucleus, like I told you, contains DNA. And DNA is made up of, you have the chromosomes, which are structures within the nucleus that are made up of DNA. So think about it this way now. DNA, chromosome is in the nucleus, and chromosome is made up of DNA. The DNA is wrapped around by histone protein. Does that make sense to you? Yes. Now, chromosomes are the big, chromosomes are the big structure in the mind, but the chromosomes are made up of DNA. A stretch of DNA, a particular segment of DNA makes a gene. So chromosome, made up of DNA. Let me get out a marker. I usually use a marker when I'm teaching this. Now, this is the DNA. This is the chromosome. This side, which is different, I'm going to cut it off now. This is a gene. This gene can be coding for your eye color, but there are some DNA that makes up the gene. So a stretch of DNA or a segment of DNA makes up a gene, and the gene altogether makes up a chromosome. So there can be another gene here, there can be another gene here, there can be another gene here. Now, the location of a gene on the chromosome is what we call a loci or a locus. All right. Now, so chromosomes are usually, if you notice that chromosomes are usually drawn like this. It tells you that they have two strands. So two strands, each of the strands is called chromatid. It's not chromatin, because I'll be talking about that in a bit. There's difference between chromatid and chromatin. This is chromatid. Now, so there are two chromatids makes up a chromosome. So chromatid plus chromatid equals a chromosome. Now, but there is what we call chromatin. Now, chromatin that ends with N is when chromosome is not condensed. Now, think about it this way. If you want to sweep with a broom in your room, if you want to sweep your room with a broom, or you want to use a vacuum, before you use the vacuum, you may feel that the room is dirty, but you may not be able to pack up all the dirt. They are all in fragments, and you may not notice a lot of them as though it's a lot. But by the time you finish using the vacuum, you notice that the vacuum is full. It has a lot of dirt packed up in it already. And you may be surprised that, are all of these things in your room before? Now, what is the difference? Before you use the vacuum, the dirt are uncondensed. They are sparse. They are scattered out. But the moment you use a vacuum, you condense the dirt together in the vacuum box. And now, in the vacuum container, now you have all the dirt packed up together, and you can empty it. So chromosome is the condensed chromatin, whereas chromatin is uncondensed. All right? When it is uncondensed, you have chromatin. When it is condensed and it is visible, you have chromosome. So that tells you, from the analogy of the room, the dirt in the room, you can infer that chromatin is invisible. It's not visible as chromosome. You just see them as particles like nucleus. But once they are condensed, they become chromosome. And this usually happens during the prophase of the mitosis stages. Now, so this is chromatin, and it's a chromosome protein complex that looks like an unwound. Can you see? It is just like the way you see nucleus. They are not visible as chromosome at that time. But don't forget, the key word is they are not condensed. Then you have the nucleolus, which is what houses the cell's DNA. And again, the nucleolus, inside the nucleolus is what makes your RNA. It helps to make some RNA. It helps to it helps to make some RNA. So we have different types of RNA. You have transport RNA, you have messenger RNA, you have ribosomal RNA. So the nucleolus helps to make ribosomal RNA. It also helps to make ribosome. All right. Then you have the nuclear envelope. The nuclear envelope is a double membrane which has pores. So once some molecules are able to go out and into the nucleus as well, then it also separates the nucleus from the cytoplasm. Let's go. Now, chromatin and chromosome, I've talked about their structure. You can see this is a chromosome. This is a chromosome. And that tells you that half of this, like this, is going to be a chromatid that ends with a D. And this is also going to be a chromatid that ends with a D. So a chromosome is made up of two chromatids. And at the center of it, the location where they are joined together, we call it centromere. I'll be talking more about this when we do cell cycle, where we talk about mitosis and meiosis. So this is centromere, the location where they are joined together. And I will be talking about the protein that joined them together then clearly, but the name of that protein is called kinetochore. So centromere is a location, kinetochore is the protein or the glue that holds them together in that location. Kinetochore. Now, you can see this is a chromosome. Half of it is going to be chromatid, and when they are uncondensed, and they are not as visible as chromosome, we call them chromatin. So uncondensed chromosome is chromatin, and condensed chromatid is chromosome. I hope you got that. Now, what do we call chromosome or chromatin in the first place? It's just protein. It's just protein histone protein and DNA. So this is histone protein and DNA. They one together. The histone protein gives the DNA its structure. DNA is strand. So the histone protein is what gives it a structure, and the DNA winds around it. So histone protein plus DNA is what makes chromatin chromosome. All right. Now, in eukaryotic cell, the chromosomes are linear structures, but in prokaryotic cells, they are circular. Now, chromosomes are usually visible and distinguished from one another when the cell is getting ready to divide. Did you see that? That is at the prophase stage. That's when chromosomes become visible. That's when they become condensed. During the interphase, I'm not talking more about it now, but I'll be talking about it later. During the interphase, chromosome will only be invisible. It will appear as chromatin. So look at it. When the cell is in the growth and maintenance phases of the cycle, proteins are attached to the chromosome and they resemble an unwound, jumped branches of threads. These unwound proteins, chromosome complexes, are called chromatin. Does that make sense to you? Yeah. All right, now, so chromatin describe the material that make up the chromosome, both when condensed and decondensed. Don't be confused with that. Get the concept. Chromatin is when they are not unwound, when they are not visible. That's chromatin. When they are not packed together, meaning that when they are not condensed. But chromosome is actually when it's visible, but then it's still the same content, is the same DNA and histone protein. All right, now ribosome. Now, I've told you earlier that the ribosome is what is responsible for protein synthesis. So every cell needs ribosome because every cell needs a protein. Now, there are large complexes of protein and RNA, so they are made up of protein and RNA, and they consist of two subunits. They have the large subunit and the small subunit. Both of them are committed to protein synthesis. Now they receive order for protein synthesis from the mRNA. Now you should have heard of central dogma of biology. Central dogma of biology says information flows from DNA, it flows to RNA, so I can put mRNA here, and it flows straight to proteins. Now, if you have an information in your DNA and it doesn't flow to protein, then the information will not be functional. Now, that is where we come up with the concept of recessive and dominant. If an information is in your DNA but it is not converted to protein, we say that the information is recessive information. Whereas when the information is converted to protein, we say it is dominant. So the information that is converted to protein is what we see that is functional in your body. All right, so after completion in the nucleus, the mRNA is going to... So now DNA is converted to RNA or mRNA inside the nucleus. But once it is completed, once it produces a nucleus, once it produces mRNA, once the mRNA is produced in the nucleus, then it goes outside of the nucleus to the cytoplasm. Now, think about it. Why do you think the mRNA leaves the nucleus to go to the cytoplasm? Now, the purpose of it is this. Ribosome needs the mRNA to make protein, but ribosome is not in the nucleus. Ribosome is only located outside the nucleus, on the nuclear membrane, on rough endoplasmic reticulum, and inside the cytoplasm. So mRNA is a raw material that is needed to make proteins, but it has to get out of the nucleus to where ribosomes are, because no ribosomes, no proteins. All right, so after completion in the nucleus, mRNA travels to the ribosome in the cytoplasm, where mRNA codons is translated to the sequence of amino acids. Does that make sense? Yeah. All right, like I said, as you listen and if you notice any question, please feel free to reach out to me and I will respond to it. Then you have the mitochondria. The mitochondria, usually we call it the powerhouse. Why do we call it the powerhouse? That's what generates ATP, right? Sometimes you want to do some work and you joke with it that I don't have ATP. You're saying you don't have energy, meaning that your mitochondria is not producing what it should produce at that time. Maybe because you don't have glucose, right? And so the mitochondria is also double membrane organelle. It has a oval shape. It has an oval shape. It is double membrane. It has their own ribosome and DNA as well. There's this concept which I will be discussing later, endosymbiosis principle. Endosymbiosis principle talks about the possibility that mitochondria and chloroplast would have possibly been a prokaryotic organism that later engulfed into an eukaryotic cell. So the thought is that since they are able to survive outside the cell by themselves, that it's possible that they were once prokaryotic organisms that were engulfed that were ingested by eukaryotic cell. So they have their ribosome, they have their DNA by themselves. They are responsible for making ATP, and they also carry out cellular respiration. And what is cellular respiration? It's still the process of making ATP. You heat glucose, the cell break it down and produce ATP from it. That is cellular respiration. It's harvesting energy from chemical energy. Each membrane is a phospholipid bilayer that is embedded with protein. Now this process uses oxygen and it produces CO2 as the waste product. But we'll be talking about cellular respiration in a bit. The inner layer folds into what we call the crystal, which is what you have here, and the outer layer, it's surrounded by outer membrane as well. So the area surrounded by the fold is called the mitochondrial matrix, which is where major part of the cellular respiration takes place, which is where ATP is made. All right, I think I'm taking a lot of time now. Let me see if I can move fast. Wow, yeah, let me see if I can move fast. There are some things I will skip, not because it's not important, but because I want to save time, not to make this video too long. And I know that you can study those by yourself. Now, when you see pericytosome, just think about it. It looks like lysosome, but it's not the same. Pericytosome helps to metabolize waste. Now, what it does majorly that is very profound is that it converts H2O2 hydrogen peroxide into H2O and oxygen. Now, this is very profound for the cell because, so I'm going to have 2 here. So we have 4 oxygen now, and I'm going to put, if I put 2 here, then I have 4. Good. So this is a very profound thing that pericytosome does, and it saves the cell a lot. It is to break down hydrogen peroxide, which is toxic to the cell, and break it down into water molecule and oxygen that are friendly to the cell. So take note of that principally. That's one of the presence of that is actually a major thing that makes it different from from lysosome. So there are also small membrane-bound organelles resembling the empty lysosome, and they contain enzyme to digest fatty acid as well. They are the products used by mitochondria to make ATP, right? All right, so let's go. The vesicles and vacuoles, I want you to look at that yourself. Yeah, just read that up. Centrioles, majorly what centriole does is that they are located in the centrosome, and they are composed of microtubules. And when you think about this, think about the cell cycle process, how centrioles make spindle fiber. So majorly they help with cytoskeleton as well. They are part of cytoskeleton. They think about how they help to make spindle fiber that holds chromosome at the center and pull them back at the anaphase stage. But read up the descriptions there, please. Then lysosome, please read up lysosome as well. They also have digestive enzyme. They break down. Now, I think I should talk about this a little. Now, think about this. When you have cell heat in many ways, there are some materials that are large that cannot go into the cell. So because of that, they need to be engulfed. And the process is called phagocytosis, which is the same thing as cell eating. So the cell is going to engulf. So the cell is going to engulf food particle, like you can see here. The cell will engulf food particle, just like it is here. The cell takes off food particles. I'm trying to get a marker. So I'm not able to write. I don't know why. I could write normally. Okay, so the cell is going to engulf food particles like you see here. Then what is going to happen? The engulfed food particles goes into the cell through it's going to invaginate the cell membrane like you can see here. Then it goes into the cell. Now you have the food particle inside here in a vacuole, right? Now, what the place of the lysosome is to fuse with the food particle vacuole that you can see here. So it's going to fuse with it. Once it's fused with it, the lysosome is going to release its digestive enzyme into the cell, into the vacuole. You can see that. Then the digestive enzyme is going to break down everything and release the food or the particle to the cell. So that is what lysosome does. So please read up the process. You can see this here. They are also part of the endomembrane system, which was what I described the other time. They are part of the endomembrane system also. Now the cell wall is a rigid covering in plant cell. You see cell wall in plant cell, you see it in fungi, in protists, and in prokaryotes. What do? They protect the cell, they provide structure, and the cell wall is made of cellulose, which is a type of carbohydrate. I think you should remember that now from chapter 3. It is a type of polysaccharide, right? Do you remember that I mentioned cellulose then? Now, so what it does majorly is that it gives shape to the cell, to the plant cell, and that's why you see that the plants have a definite shape. Is because of the cell wall. So cell wall maintains the shape of the cell, of this plant cell. Now, don't misunderstand it. Cell wall is different from plasma membrane. Okay, all right. Now, the chloroplast is what you have in plants that makes it plant. The chloroplast is what takes that regulates photosynthesis. No chloroplast, no photosynthesis. So photosynthesis is a series of reactions that produce food molecule. That's what produces your glucose. So plant is going to trap energy from sunlight. Then it uses the energy in the presence of water molecules to make glucose. So it's sunlight in the presence of CO2, rather, carbon dioxide. So sunlight plus carbon dioxide, then water molecules is going to make glucose and oxygen. So you end up having CO2. Let me see if I can write that here. I'm not dealing with photosynthesis now, but let me just put that here. CO2. Then sunlight is going to come in. Sunlight comes in. That's my sunlight. Then it gives you 6H12O6. This is 12 here. And it gives you O2 too, which is oxygen. You have 6 here. You have 6 here. Chemical reactions should always be balanced. You have 6 here. So that is your glucose. And this is photosynthesis. All right, so the chloroplast have an outer membrane. It also has an inner membrane, as we mentioned earlier. Now, it also has inside of it some membrane structures that we call thylakoid. And the thylakoid is what you can see here. This is 1 thylakoid, this is 2 thylakoid, this is 3 thylakoid. Now, there are 3 thylakoid here. And think about it. It looks like a slide. It looks like a layer. It looks like pancake that you put together. So think about it. You have 3 pancakes here. You have 3 thylakoids here. These stacks makes what we call a granum. So the stack of thylakoid makes a granum. Now, if it is more than a granum, like this now, we have 1 granum here, we have 2 granum here, we have 3 granum, which is made up of 5 thylakoid, we have 4 granum made up of 2 thylakoid, we have 5 granum here. Now, if it is more than 1 granum, we call it grana. So opposite of granum is grana, G-R-A-N-A. Now, so it's the same thing. It's just that, not opposite, plural, right? So 1 is called 1 stack of thylakoid, not 1 thylakoid. A thylakoid is 1, right? Like it is a pancake, like a slice. Then 2 or 3 becomes a stack. The stack is called a granum. More than 1 granum is called a grana. Now, the space in the thylakoid membrane, we call it the thylakoid space. Those are a few things I should just point out. We call it thylakoid space. Then the light harvesting reaction takes place in the thylakoid. So the thylakoid is actually what contains the chlorophyll that attracts light reaction. And the synthesis of sugar takes place in the fluid side of the inner membrane, which is called the stroma. So food is made, the sugar is actually made in the stroma, which is here, right? This is the stroma, the fluid here. Then the thylakoid contains the pigment, right? I talked about that, which captures light and drives photosynthetic reaction. So I'm going to run away from that. Endomembrane system and protein. I think I will try to look at this. I've talked about this earlier, but I will just make a little discussion here before I run from it. All right, now the endomembrane system is the endoplasmic reticulum. Okay, did I say endo? Okay, so the ER is endoplasmic reticulum. It can be smooth, it can be rough, and actually is a continuation of the membrane of the nuclear membrane, like I mentioned earlier. So it consists of membrane tubules and flattened sacs. Now we have this rough and the smooth. The smooth does not have ribosome on it, and so since they don't have ribosome, they don't make protein. Presence of ribosome is what tells the function of if it makes protein or not. Anywhere you see protein, ribosome, it makes protein. That's what it does. So proteins are made by rough endoplasmic reticulum, but the smooth only made lipids. They also help with detoxification. Let's go. Now, Golgi apparatus consists of a stack of slightly curved flattened circle. Now, what do? Now, one side of it, so Golgi apparatus have two sides. It has a cis side, which you see here, and the trans side. The cis side is where it will receive packages or vacuoles from the endoplasmic reticulum. So once endoplasmic reticulum makes a protein, or when the nuclear membrane makes a protein from like that, right? You remember the nuclear envelope, the endoplasmic reticulum is a continuation of the nuclear membrane. So once they make a protein, it packaged it into vesicle. The vesicle is going to be received at the cis face of the Golgi apparatus. Then it will stick to it, engulf it, invaginate, and it becomes, it glues to the membrane. Then it continues. So the protein would later be processed in the Golgi apparatus. Once it is processed in the cisterna of the Golgi apparatus, then it's going to leave the Golgi apparatus. It's going to be packaged into another vesicle that will leave the Golgi apparatus, that will later be stuck to the plasma membrane and then get out of the cell. That is what we call endomembrane system. All right, so majorly, like I said, the cis side is towards the ER, then the trans side. Trans, think about cis. Cis is receiving side, then trans is transportation side, right? So think about it that way. And majorly, what does it do? The Golgi apparatus receives, it processes and package protein and lipid so that they may be sent to their final destination. Like I told you, the proteins that are made from this rough endoplasmic reticulum or the nuclear membrane, they are going to be packaged to be sent out of the cell. Then the cytoskeleton, it maintains the shape and assists with movement. All eukaryotic cells have a cytoskeleton. It's a network of protein fiber within the cytoplasm. It's used for support and also for shape, right? The fiber can assemble and reassemble. So just look at that. Majorly, they help to anchor organelles in place. Like I told you, cytosol is a gel-like solution, and it is the cytoskeleton that helps. Now think about it. Cytoskeleton, cell skeleton, is a skeleton of the cell. All right, so it does the same function that your normal skeleton, your body skeleton does too. And we have three types of fibers. We have the microfilaments, we have the intermediate filament, and the microtubules. The microtubules are usually the largest, the intermediate is in between, and the microfilament is the smallest. So think about microfilament. Feel like small, like it's just filament is small, but the tube, they are big, right? Like you can see them here. These are the microtubules, they are bigger. The microfilaments are smaller. Intermediate filament is in between. All right, I'm just going to skip the microfilament. Please study them. Watch the YouTube video I have there, the intermediate filaments. Just look at that up. This video is already 1 hour and I want to make it shorter. So please look at this, study this by yourself. Yeah, this is something we've discussed. This is just a review of what we've discussed. Yeah, so please just study the part that remains yourself so that I don't take it too long. Please make sure you study the gap junction, the desmosome, the tight junction. Yeah, please look at that very well and be able to differentiate them. I have to stop this now. Have a good one. Please feel free to send me questions if you have any at any point.